In this video, we will discuss about this MCL-1 antagonism will enhance the anti-invasive response in combination with Dasatinib of pancreatic cancer cells. So, in the previous video, they have checked many patient-derived cell lines, and in that case, they are able to see that one cell lines, that is TKC005, and that is TKC005 has high expression of MCL-1 and also high expression of this SRC Y416 was phosphorylated SRC. So this is chosen to be the candidate where they will see the impact of both this MCL inhibition and SRCC inhibition. And for the SRCC inhibition, they use Dasatinib. And so first they check the impact of MCL antagonism in a dose-dependent manner on the MCL concentration, and they are able to see with the dose-dependent antagonisms of MCL, they can see the increase in the MCL concentrations, indicating that there is some counter response or feedback response that will cause more expression of this MCL1. And when they check it for the different time intervals, that is 24 hours, 48 hours, 72 hours, they are able to see the coffin expressions goes down with MCL1 antagonism. Total coffin expression goes down with MCL1 antagonism, but this phosphorylated coffin expression increases with time. When they check its ratio, they can see that total coffin goes down in the case where MCL1 antagonism is used, whereas when they check the ratio of this SC coffin and total coffin, they see that it goes up, indicating that the activated coffin is increased with time in the presence of MCL1 antagonism. And when they check for the MCL1s, since that these results are similar to what we have seen in the panel A, that with a MCL1 antagonism treatment, they see more MCL1 expressions compared to the control case or vehicle case. And similar to the results that we have observed for the total coffin, we have seen the results for the total SRC. Not similar results, there is some difference, that is total SRC does not get affected whether the MCL1 antagonism are present or absent. But when we see the activated SRC, that is phosphorylated SRC at Y146, they can see the phosphorylated SRC goes down, indicating that the MCL1 antagonism somehow suppress this SRC, the activated SRC with time. And thereafter they check the synergetic effect of the Dasatinib at different concentration and this S63845 at different concentration, and they are able to see a synergetic effect in most of the regions that is highlighted by this dark blue color for the different concentrations of Dasatinib and S63845 at 48-hour time intervals and at 72 hours time intervals. We can see that the large portions of the region shows the synergetic effect. And then they check the invasion properties, apoptotic properties, and this proliferative properties using Ki markers and this apoptotic markers and Ki61 markers, apoptotic markers, and in the immunohiston of the samples and at different conditions, and they do not see any too much significant difference with the dose dependent increase in the Dasatinib or dose in combinations with A121047, which is this MCL1 antagonism. But when they check it on, so this indicates that the local invasions does not get affected, but when they check the overall impact, they are able to see that when this Dasatinib and MCL1 antagonism are used together, they are able to see a decrease in the invasions with the dose dependent increase. They are able to see decrease in the invasions at around 10 macromolar concentration of dasatinib and also for the apoptosis, and a continuous decrease in the invasions with the dose dependent increase in the dasatinib and MCL-1 antagonism. And this is the panel I is for the invasion properties, and panel H is the grid properties. So you can see that it's indicates more of like local invasions, and they do not see too much impact on the local invasions for both proliferative and apoptotic, but the invasions index shows that there is a decrease in the invasions with dose dependent increase in the dasatinib and MCL-1 antagonism. And these results are in line, the invasion results are in line with this invasions properties. So this indicates that the local invasions does not get too much impacted, but the check the bigger area there or invasions properties, they are able to see a decrease in the proliferations and apoptosis at around 10 macromolar dasatinib, or dose dependent also they are able to see a decrease in the apoptotic factors for and also for the invasion properties. So this all in together indicates that both these MCL-1 antagonisms in combination with dasatinib has some role towards this anti-invasion properties or suppression of invasion properties of these pancreatic cancer cells. So I hope you got some idea what is going on in these experimental results. If you need to understand more about these experiments, you can go through the reference that has been provided in the description. Mainly you might have to go through these results of the H and I again, so that you will understand this thing in a better manner. That's all for this video. Thank you for your time.